First, a calcium loading solution is prepared, warmed, and bubbled. The urinary bladder is dissected out, opened, pinned out flat, and a strip of detressor is taken. The detressor strip is added to the loading solution, incubated, and rinsed. Pinned out flat, the calcium fluorophore within the tissue is excited by laser light and the emitted light captured. Resulting movies consisting of individual frames are analyzed by comparing successive frames. The background is subtracted, the cell edge threshold determines where events can be found, and a ratio between successive frames is taken. If the remaining events match predefined criteria, they are highlighted and labeled.
Hello, my name is John Young from the laboratories of Keith Brain and Tom Kinane at the Department of Pharmacology, University of Oxford.
I'm Rob Amos, also from the Brain and Kinane labs.
And my name's Keith Brain. Today, John and Rob will show you a calcium imaging technique in smooth muscle. We use this in our laboratory to study the release of neurotransmitter from autonomic nerves and its effect on smooth muscle.
So let's get started.
The calcium indicator, Organ Green 488 Bacta 1AM, comes in 500 microliter vials containing 50 micrograms of powder. To prepare the calcium indicator, add 40 microliters of Pluronic F127 solution to the powder and shake gently for 30 seconds. After that, add 460 microliters of physiological